Okay, can everybody hear me okay? So I'm a little bit different speaker in this audience today, but that's okay. I work in a quantitative genetics group, which is known as the Center for Genetic Evaluation of Livestock. That's a large group at Colorado State. But the reason I work there is I'm a DNAologist. I was really trained as a physiologist and molecular biologist, and so as the DNA world has progressed, my world has become quantitative genetics, cattle breeding, and so forth. I'd like to make just a couple points before I dive off into all the information we're going to talk about, and building on what Derek said. The first point is this: DNA is really a pretty simple thing, nothing more than A, G, C, and T. And the ability to computerize that is what moved us so quickly with this technology. And to give you kind of an example of that kind of technology explosion, how many people got like a smartphone in your pocket, right? Well, that's nothing more than ones and zeros. That's how it works, right? But it's the processing of the ones and zeros and having that in a computerized format that makes a smartphone. Well, that's why DNA technology has moved forward like that, because the biochemistry of it is so simple, A, G, C, and T, and then the reverse complement of that has really made this technology explode to where we can do it in computerized ways and do it so fast. So now we're at the point where when you get to what some of the simple things that we've known for 100 years about principles of genetics, we don't have to talk about like big B and little b anymore to talk about coat color, right? Everybody knows that, right? Big B is black, little b is red, and big B is dominant, that kind of scenario. We don't have to do that anymore. We know that is a color mutation in the bovine. It's on chromosome 18. It's in the melanocortin gene, and it's a GT SNP. We know that. So we don't have to use big B, little b anymore. We can actually use the alleles, G versus T, and that's the SNP, okay? And so we're really at a point now where we're starting to talk about traits, phenotypes, at very, very specific at the genetic level, but it's not just genetics of sire and progeny. It's actually what is the sequence, okay? And so that's where my talk is going. Hopefully I'll give you some background into understanding, our ability to genotype and how we can do that so massively today. But I will tell you, the challenge to move a project like this forward is not DNA anymore. It's not genotyping, it's the phenotype. That's our challenge. We can do the genotyping at unbelievable levels. Okay? Our challenge is the phenotype. Okay? So hopefully when I get through with that, maybe somebody will buy Derek a new horse, whatever, more GPS collars, but that's our real challenge. So let's just talk a little bit about genetic improvement. We can do genetic improvement. If you can measure it in parents and progeny, we can do it. So this is just a slide I like to show. Here's a turkey carcass at 112 days of age and 196 days of age. That's what a turkey carcass looked like in 1966, and this is what our modern turkey carcass looks like. So if you're like me, I'm not a big turkey fan. I like it one day a year, Thanksgiving. But if you're like me, I don't like to eat turkey for 2 weeks after Thanksgiving. You know who's to blame for that? Geneticists. Geneticists are to blame for why you have to eat leftovers for 2 weeks after Thanksgiving. Because look at the difference that's been made. Selection for massive breast size of the turkey has created a beast, right, that has to be bred artificially because the tom physically can't make the hen anymore. That's because of genetics. We created them that way. Think about that. How many turkeys do we need on Thanksgiving Day to feed the United States? It's a bunch, okay? And so that's what genetics has done. I'm not arguing whether that's good or bad. I'm just saying that that's what it is. Now, this is a classic graph. When we talk about genetics, a lot of our old world genetics, unless it was something really simple like black and red, polled and horn, a lot of it has been done with quantitative techniques. And when I say quantitative, I mean, it took at least 500 animals of very, very good phenotypes and pedigree to measure what we call the heritability. And so I want to spend a little time on this slide. So what we here have two different traits. This is what we would call an extremely highly heritable trait, heritability of 0.7. And down here we have a bunch of graphs of what we call lowly heritable trait, a heritability of 0.1. And so what this means is that 70% of the phenotype in that trait there was due to genetics. Down here, only 10% of the phenotype is due to genetics. So let me explain it. So here we got the line, that's the mean of the population or the average, and then each one of these little clusters of graph represents an animal. So this animal was a little bit above average. Well, the reason it was a little bit above average because what is white was the environmental effect. That animal was raised in a favorable environment. It actually had a negative genetic basis or genetic variance in this case. But because the animal was raised in such a good environment, it was above average. Okay? And so what you should really see, the difference between this set of graphs and this set of graphs, is the size of the white bars. Okay? So if it's highly genetically regulated, 0.7, then the white bars are not very big. Okay? But if it's highly environmentally influenced, the white bars are huge. And actually most of the, when we talk about livestock animals, and particularly animals that are out in environments, I'm not talking about pigs and chickens that are raised in confinement buildings, the white bars are really big. And so we really in livestock, even though we might say we have a highly heritable trait, what we call highly heritable is really not that high. So if we have a trait where 40% of the variance is due to genetics, that's huge in animals. So that means 60% of it is still due to environment. Okay? And I think when we get into the DNA world, and I get frowned upon sometimes, particularly by my colleagues that work for DNA companies, because, you know, I come to this thing and I say, well, you realize that that DNA test didn't really mean that much because most of the trait is environmentally influenced. Well, that's true. These are live animals. Okay? So when we talk about DNA technology is going to do a lot for us, but we got to keep it in perspective. It's not the only part of the story. So I like to lay that out at the beginning before I go and talk. Okay, Derek started talking about EPDs, and you know what EPDs do for us is we can rank the animals, you know, they give them some kind of plus or minus value. So if we're looking at animals for birth weight, if a bull is +5 for birth weight, well, we probably don't want to use him on heifers because that means his progeny are going to be 5 pounds heavier than some base, and that could be pretty nasty, you know. So if 85 pounds is our average and 5 plus makes them into the 90s, that not be very good for heifers. But if we go to -5, that might be a very good heifer bull because if 85 is our average, that means the calf should be around 80 pounds. You know, that could be very good. And so there's been a real evolution over about 30 to 50 years of being able to calculate EPDs. You know, in the early days, you know, we used pedigree and then it builds on itself. Now we use multi-trait models. Now we're using multi-breed models. And now we're taking the genomic information, the DNA information, and putting that into the EPD. Okay? And what really is important here is this term here. We start out when an animal is a yearling and he doesn't have any progeny, his EPD is very inaccurate. But then as he gets more and more progeny, this information becomes a lot more accurate. So in the dairy industry, there are Holstein sires out there that have over 1 million granddaughters in production. So with that kind of data, we can say, well, heck, we know pretty much who's the big milkers and who's not the big milkers in Holstein cattle. You know, in beef cattle, there's a few examples like EXT, that was a famous Angus sire. You know, he put like 10,000 daughters in production in the U.S. and maybe another 5,000 in Australia. That's pretty good. I mean, we know what EXT is all about. I mean, he's kind of a big growth sire, and his data, you know, his EPD information is pretty accurate. But when we start talking about beef cattle and difficult traits, we've got to realize that we're never going to have that kind of data. So DNA information can help us a lot, take us forward, but we're never going to be in a situation of selecting beef cattle like we do with poultry or pigs or Holsteins. We're just never going to have that data, okay? But we could get a lot better. All right, so here's information I want to share about kind of the status of where we're at with genomes. What do we really know about the DNA of different species? Okay, so up here I have cow, sheep, goat, and the yak. So we have a full genome sequence on all of those animals. And the first bovine that was sequenced was an inbred Hereford Dominette out of the Miles City USDA ARS breeding program. And the reason she was the first bovine that was sequenced, and we spent $50 million to get that, not much more than a little less than 10 years ago. And the reason we did is because she's so inbred, she's very homozygous at almost all of her loci. Okay? And so her sequence is very valuable because we can compare all kinds of other animals to her and see where the differences, the mutations and SNPs and things like that occur. So there's a big group of scientists of us that work together in what's called the National Animal Genome Program. That's actually instead of a regional project like a lot of other people are members of, like I'm a member of the W1, that's the Western Region Beef Cattle Breeders Group. We work on a national scale to make decisions about things like why should we sequence Dominant, okay? And you can go to animalgenome.org, that's a really cool website that has all the current information, you know, about what's been sequenced, okay? But since Dominant was one of the first ones that was sequenced, the other technology that has come beyond that has just been, has rapidly come forward. So additional animal sequence, the first bovine sequence cost us $50 million today. We can sequence a bovine for $5,000. Actually, we could do you for $5,000 if you would like to have that information. Okay, I don't know what you would do with that if you had it, but you could get that done today. And then since we have all that sequence, now we're making SNP chips. The first bovine SNP chip that was made was what we call a 50,000 SNP chip, and it has 50,000 genotypes on this chip. And so what it does basically is it just starts at the top of chromosome 1, and then there's a marker about every 40 kb as it walks across all 29 autosomal chromosomes and then the X and Y. So there's 30 chromosomes in the bovine. Well, that was pretty cool, and we're going to show you some of that kind of data. But now we're moving up to a higher level. So now we have an 800,000 K chip. And so now it's just like a ladder going across each chromosome. Instead of being 40 kb, now we're down to, there's a SNP or a marker every 4 kb. Okay? But actually, you know, in the bovine world right now, there's over 10 of these SNP chips on the market. Okay? So you just start to think about this. I did my first 50K genotyping project. We genotyped 800 heifers for a fertility project, and we had a little over 47 million genotypes in that data set. Okay? So again, the issue here of making genetic improvement is not about getting the genotypes. We can generate genotypes, whole genome sequence at an unbelievable rate today. The question is, how do you get the phenotypes and put it all together? Okay, so just this little pictorial about what are people doing when they get all these genotypes. Well, this is a SNP chip. And each one of these wells, we would put the DNA of one animal, and this one is a 50K chip, so we would get 50,000 genotypes from this animal. And then we would do statistics between the phenotype and the genotypes, and we call that a GWAS, genome-wide association study. It's just a simple genotype to phenotype association statistically. Okay? If we use that information to actually make breeding decisions, then we would say we're doing whole genome selection. So that's an acronym that's known worldwide of anybody working in genetic improvement today, whether you're talking about plants or wheat or cattle or whatever, you're doing WGS, and then you would take that information to make a better breeding value or an EPD. Okay? Well, that works out really, really well if you have a situation like Holstein, where everything, you know, the animals are in the same kind of pens, they're eating the same kind of feed. I mean, that system really works well. But when you start talking about very, very difficult traits like grazing distribution, then, you know, maybe we don't have clean data sets. So then knowing the genes, okay, could even be more valuable than just a whole genome breeding value. And so when you start talking about traits like we've been talking about in here today, well, we're never going to have a sire with 1 million granddaughters. Knowing the genes and what regulates the physiology could be extremely valuable in us making tools that can improve the trait. Okay, so I want to go through this slide. This is of Holsteins to make my point. Okay, so what we have here is these are the common traits that we talk about in dairy cattle. Okay, and then we have three breeds of dairy cattle: Holstein, Jersey, Brown Swiss. And of course, as we know today, I mean, you can drive around the earth. Maybe you can find a few other colored breeds of dairy cattle, but primarily in the world today, if it's milk cows, it's Holsteins. Okay, and you know, they're a tremendous beast. Genetic improvement has doubled the amount of milk produced by a Holstein cow in a 305-day lactation in 50 years. Okay, more than doubled it. Okay, and so we have tremendous pedigree knowledge of the Holstein. Actually, the U.S. Holstein is a little more than 5% inbred. We know what the good bloodlines are. And because of that, all of that knowledge, clean data set, if we produce a Holstein bull and we're considering if, you know, he should grow up to be an AI sire, the minute he's born, just by the pedigree and the quality of that data, we would say we're already at an accuracy level or a reliability level of 35%. Okay? Well, so then the process for many, many years of determining whether a Holstein bull calf was going to grow up to be an AI sire was they had to go through a sire testing program. And the sire testing program, though, took 6 years. Okay? So what I had to do is you have to grow up that bull, and he has to get to the age you can collect him free semen. Then you have to take some of his semen, you got to go breed some cows and make some daughters. And then you got to grow up those daughters to the point that they could get bred and then have their first baby so you could have their first lactation records. The process took 6 years and anywhere from $75,000 to $150,000 to get a proof on a Holstein bull. Well, now we have DNA technology. And actually, there's probably been over 100 Holstein bulls sequenced, full complete sequence. There's over 60,000 Holstein bulls, I mean prominent AI sires with complete high-density genotypes, and they take all that data and put it together and made this table. So this is very interesting. This table was actually made before 2010, and so what I want to show you is let's just focus on this trait right here because this is just easy math. Percent fat, right? That's percent fat in the milk. How much fat is in milk? Well, it's about 4%. If you buy what my wife buys, she buys the 2%. But that means it's been processed, right? Okay? So what this means is because of the DNA technology, the minute that bull calf was born, we were already at the 35% level of accuracy because of the pedigree. But now because of the DNA knowledge that we have, okay, you can actually add those two numbers together. And so the moment that calf was born, we genotyped the calf, and we know the breeding value of the calf at the 80% accuracy level. That's tremendous. I mean, that's a woohoo moment for ag scientists, you know. You know, saying, hey, look what, you know, we did research and we impacted the industry. That's really a translational effort. But now let's talk about that a minute. So we're talking about Holsteins, right? And yeah, we got that phrase, happy cows in California, but you know, they like to show them grazing out on green hillsides, but we know what Holstein production's about. Holstein's a very healthy animal today. We raise them in concentrated feeding operations. You know, it's a very, very defined, very focused food production business, right? The data is very clean. It's one breed, not 10 to 80 breeds, however you want to count beef cattle. And the traits, it's very, very goal driven. What's the trait? Milk production and 4% fat. That's the traits, right? So it's very focused, and so the process worked very, very well. The translational process of taking DNA information and putting it together, let alone just the size and scope of the data set, right? Okay, out of time. Okay, so when we talk about beef cattle, though, right, it's not such a clean world. I'm short on time, but when we talk about beef cattle, here's some of the initial genomic EPD information. Look at the accuracy level. It's very low. Why is that? Well, it's because we got multiple breeds, multiple traits, different goals among breeders and so forth. So it's very, very difficult, right, to make that system work in beef cattle the way it has worked in Holsteins. So when we talk about DNA technology, now I'm going to talk on both sides of my mouth. It's going to be very, very difficult to do this in beef cattle. However, though, what I see as the future of DNA testing is going to be in the difficult traits. Why? Because there's no other way we're going to get these measures, right? Grazing distribution, how many cows can we collar? What's the price of the collars? DNA technology can give us a lot. Is it going to give us accuracies like Holsteins? No way, it's never going to happen. We're never going to have that kind of data. But will it give us breeding values? Yes, it will. What's the value of those breeding values? Time will determine, but I will tell you this, and I'm going to be a little terse here, so get ready. Those breeding values are a damn sight better than ignorance, right? So we'll on to the next slide. So I've been doing a lot of research now. I worked at New Mexico State 15 years. Now I moved to Colorado, but it's still a very, very dry place. So we're challenged in harsh environments with traits like grazing distribution. Even though now I'm in New Mexico and we all talk about high elevation, but I think if you went back to the last graph, the dry areas, the high areas, they're very synonymous areas. And so Derek showed these kind of slides and talking about hilltoppers and bottom dwellers, and we're collecting DNA and genotyping animals. Get to this. So this is the graph where Derek gathered his information to talk about how much variation is being accounted for by the SNPs. This is a classic kind of graph. Anybody that does genomics, whether you're working humans, plants, dogs, cats, cattle, whatever, we make these kind of graphs today. This is called a Manhattan plot. The reason it's called a Manhattan plot is because if you look across the top of it, it's like the Manhattan skyline. It's just a lot of ups and downs, Okay? And so what we're really looking at here is on this axis here are the chromosomes. Okay? So this is bovine, so there's chromosome 1, the autosomal chromosome 29, and then the X. And then on this axis is the significance. So the higher the dot, the more significant. And so when you get a dot that's up to the top of the page, and this is the one that Derek was talking about, that's a SNP on chromosome 29 up at the top, you say, whoa, boy, there's something right there on that chromosome that is influencing the trait of rough. Okay? Sometimes this thing works, sometimes it doesn't. Right there, that's the trait. So Derek went over all of that. And so that's one of the areas that Derek was talking about, chromosome 29. Okay? So because we have the ability to sequence, and because now that we have sequence, there's people that are putting these huge databases together of what is there on chromosome 29. And so we went to that region on chromosome 29 and we said, okay, well that's pretty cool, but that's just DNA. But what does that DNA code for at that region? So DNA makes an RNA that codes for a protein, and that makes the physiology and the phenotype of grazing distribution. Well, it just so happened at that specific locus, okay, there is a gene. And the gene is called GRM5. That stands for the glutamate receptor metabolomic form number 5. Okay, glutamate receptor metabolomic number 5. And that's actually been a gene that's been studied extensively in humans and mice as a regulator of appetite. Okay? So, what does that mean? Cows walk further because they're hungry, or cows are satiated and they walk further because they're bored. You could test either hypothesis. But what I'm showing, what I want you to know is it's an exon 4 of this gene, and it was right here where this blue line is, okay, is where that SNP was on the chip. So it's in a coding region of an important gene that regulates appetite. Okay, so we said, okay, well that's pretty cool. We found it on that chip, but can we find that SNP in other animals? So what we have here is, this right here is just a mock-up of the gene that we pulled off of the Bos Taurus genome, Dominant, and then we added some other pieces of sequence information to this. So I had been doing a heifer study where we had sequenced all the RNA in the hypothalamus, because the hypothalamus is your tissue in your brain that regulates appetite. We had sequenced all of that RNA, okay? And so what we have here is green is the forward strand and red is the reverse strand, and we have many, many copies of that gene, okay? And our software said, wow, there's a SNP in that gene right there, okay? And so that was 8 heifers, and these heifers were selected on prepubertal and postpubertal. That's why we did the study. Well, he said, well, that's pretty cool. Those were Brangus heifers. Could we go to some other genomic resources? So at the Meat Animal Research Center at Clay Center, Nebraska, they had just finished a project where they had low density or low level sequenced 96 prominent bulls. 96, okay? Because we're now in the realm where we have over 1,000 bovines that have been sequenced, okay? And so we said, from that DNA information in that 96 bulls, did you find that SNP? And they said, yes, it is. It's right there. And actually, there's another SNP right here, and it was there. But then there was one more that we didn't detect in the Brangus heifers that they detected in those 96 bulls. Okay? So what we're starting to tell you here is we're getting the ability. Here was a spot on a chromosome that's regulating whether cows, are hill tappers, bottom dwellers. It happens to be in a gene that regulates appetite. And we're starting to get to the point where now we could see that SNP. So if we were going to make a tool, a genotyping tool, to determine whether the cows are going to walk further or climb higher or whatever, I would say, Dr. Bailey, we better darn sure put that genotype on there so that we could know whether the animals have this allele or that allele and which ones are favorable. So we're making some steps. So, simple conclusions, we've done some GWAS, we've done some sequencing, and we've got a good, start on starting to look at what are the actual genetics that regulate grazing distribution. And when we really start to put all this stuff together, being able to put the sequence together with the gene and all that, that really makes a cool story as we understand physiology and so forth. So just to wrap it up real quick, I work in big teams. Any kind of genomics project requires big teams, from people that do the phenotyping to collecting the DNA to doing the genotyping, doing the statistics, and all. It takes big teams. So in this case, for this project, Derek and I worked with Juan Medrano and Gonzalo Rincon at UC Davis. This project, you know, as Derek talked about, the grazing part of this has been funded by the Western Sierra, but all the sequencing project, those come from different AFRI grants that I've been part of, a couple different endowment projects that I work with, and also CSIRO is also been involved in our sequencing efforts. So, you know, I'm more than happy to answer any questions. I wanted to leave it with this. So my wife and I, we like to drive around and do wildlife photography. So, you know, here's some of these bottom dwellers and riparian area grazers. And then if you look close here, you know, these are some real hilltoppers here. This guy here, and then you see this ewe here, we got her in action going up the side of the rock face there in northern Colorado.
Okay, we got some time for a couple questions as we're swapping out audio-visual equipment, so go ahead.
Sam?